Hello, my name is Sophia and I'm a dietetic intern completing my rotation with Miss Linda. Today I'm here to speak about the benefits of exercise in regards to glucose metabolism for people with type 2 diabetes. Some of the terms that we should get familiar with are GLUT4, which is a glucose transporter in a cell that is transported to cell surface in the presence of insulin signaling and other signals from muscle contraction. Glycogen is a multi-branched polysaccharide form of glucose used as energy storage. Mitochondria is a part of a cell that is in charge of making energy for any cell as need arises. It makes it in the form of ATP, which is known as adenosine triphosphate. This is a picture of a cell with all its organelle, and we're going to focus on mitochondria, which is this little one here. And during the presentation, I will mention cell surface, which is this area right here. These are a lipid bilayer that contains all the organelles in charge of energy production, your RNA or microRNA productions, and all the protein productions. Basically, mitochondria is also known as the powerhouse of a cell. Mitochondria produce the energy necessary for cell survival and its functions. Through a series of chemical reactions, mitochondria break down glucose into an energy molecule known as ATP, which is used to fuel various other cellular processes. As we continue on terms, the term phosphorylation basically means transfer of phosphate molecule from one protein to another. ATP is the energy molecule for living cells, and it works with enzymes to release energy as it loses one phosphate group from its three phosphate group molecule, one at a time. Cyclic AMP, or just adenosine monophosphate, is a ATP without two phosphate groups. It is a signal molecule for glucose as glucose is available, and it signals the production of ATP. This picture over here, as you can see, there is adenosine molecule right here, the two cyclic figures with a ribose group and three phosphate groups. The three phosphate is the reason why it's called triphosphate. As ATP turns into ADP, it loses a phosphate group and also some energy. And as you can see, ADP, adenosine diphosphate, only has two phosphate. This continues to degrade to AMP. And finally, the cyclic AMP is the signal molecule for glucose influx and the production of ATP. On the reverse, as phosphate group is added, it requires energy to add the phosphate to the adenosine, the molecule itself. Types of glucose uptake. We have insulin-stimulated glucose uptake. Insulin is used as a signal molecule at the insulin receptor site that leads to glucose transportation into the cell. And with people with type 2 diabetes, this mechanism is impaired. The second type of glucose uptake is exercise-stimulated glucose uptake. And exercise activates molecular signals that bypass defects in the insulin action of the skeletal muscle. In type 2 diabetes patients, this mechanism is normal. So it works as the alternative that could be used to manage glucose clearance from the blood. So this is a diagram of insulin-stimulated glucose uptake. Insulin signaling involves rapid phosphorylation of the insulin receptor site on the tyrosine residue, and the activation of phosphatidyl inositol 3 kinase, which is right here. Basically, as insulin comes to the receptor side, the phosphorylation event takes place. This, along with other phosphorylations, gives a signal to the GLUT4 right here to travel, or a request is made so that it goes to the cell surface. And it goes with, it needs other transporters to take it with it. So this phosphorylation event leads to other phosphorylation and other proteins being activated to take GLUT4 to the cell surface. And many GLUT4s go at the same time because there's multiple events happening. Multiple phosphorylation event leads to many GLUT4 movements to cell surface for glucose uptake. So this is just picture of one GLUT4 going. In reality, you'll have multiple GLUT4 coming to the cell surface because there's multiple insulin receptor being phosphorylated. The glucose journey. So after the GLUT4 goes to the cell surface, glucose is allowed in. It has two mechanisms that it will go through. It will either go through to make ATP because there is a adenosine monophosphate available as a signal. So the mitochondria will start making ATP. Or the other signal that signals such as sufficient ATP will let the cell know that glucose is ready for storage. So once AMP concentration decreases in the cell, there is enough ATP, the cell slows down its production of ATP. And now if there is enough glucose still coming, it will make it into storage molecule, which is glycogen. The exercise mediated glucose uptake. This method is non-insulin related glucose uptake through GLUT4. The proteins such as AS160, TBC1D1, and RAC1 are targeted through muscle contraction, and it stimulates GLUT4 movement to cell surface. So again, GLUT4 translocation takes place. But in this form, it doesn't need any insulin. It doesn't need the phosphorylation event. It needs a muscle contraction to signal these two proteins right here. As muscle contraction happens, we also see an increase in calcium, and we see an increase in AMP, adenosine monophosphate, because ATP will be starting to get used. And GLUT4 will be signaled to be taken to the cell surface, to the lipid bilayer, so it could get more glucose inside towards the mitochondria to make ATP. The AS160 protein, it regulates insulin-stimulated GLUT4 translocation. It is phosphorylated on 6 different phosphor AKT substrate sites in response to both insulin and exercise in skeletal muscle. So this is really nice to have because in presence of insulin, this is still working to help GLUT4 translocation. And in absence of insulin or phosphorylation, your muscle contraction causes the GLUT4 action. Studies have shown that AS160 phosphorylation is increased after prolonged exercise in both humans and rats. Diabetes. People with type 2 diabetes have been reported to have impaired insulin-mediated glucose uptake, but they have a normal exercise-mediated glucose uptake. People with type 2 diabetes have been reported to have smaller, damaged, and/or dysfunctional mitochondria, including skeletal muscle mitochondria. Note that it is unknown if mitochondrial defect precedes type 2 diabetes or vice versa. Some complications and comorbidities of diabetes are hyperlipidemia, neuropathy, chronic kidney disease, cerebral infarction episodes. Diabetes also causes complications that could lead to amputations, infertility, many forms of cancers, and a decreased quality of life. Since we are so familiar with diabetes, one of the thing, it seems like a cycle when we see a decreased quality of life because it's all interconnected. There might be a few different disease states that might be what's going on together for people, patients with type 2 diabetes. Well, we have good news. According to the article, Exercise in Type 2 Diabetes, Molecular Mechanism Regulating Glucose Uptake in Skeletal Muscle, I have provided the link to this website. We see that endurance exercise training has been shown to increase mitochondrial content and activity. The study examined the effect of exercise training in people with type 2 diabetes and determined if exercise could restore mitochondrial content and function and insulin sensitivity in patient population. This study involves 14 people with type 2 diabetes. 8 of these subjects underwent HIIT training for 2 weeks, which is high intensity training, where they trained on a cycle ergometer. Each session consisted of 5 minutes warm up, 10 60 seconds of cycling at 90% maximum effort, and with 60 seconds of recovery in between, and 5 minutes cool down. You could say this was done 30 minutes 3 times a week. The benefits of exercise to type 2 diabetes and the result of the research was that people with type 2 diabetes had a marked increase in skeletal muscle GLUT4 expression. The results showed a significant decrease in blood glucose concentration. The results showed citrate synthase activity, which also significantly increased after 2 weeks of HIIT training. Now, citrate synthase is something that is used to measure the functionality of mitochondria. The research result also showed that the defect previously observed in skeletal muscle mitochondria activity were completely negated, which led to the conclusion that mitochondrial function was correlated to improve insulin-stimulated glucose disposal. So basically, because of the citrate synthase that they took the data from, they concluded that you have better glucose disposal from the blood because the mitochondrial function was improved, hence better ATP production. And I'm assuming that it also leads to having an easier exercise session compared to when you first start, because your mitochondrial function gets better, it's healed, and you have better ATP production, which means more energy when you do go and work out. Conclusion of the research is these data indicated type 2 diabetic patients increase glucose uptake and improve mitochondrial function and improve whole body glucose metabolism and insulin sensitivity. The study indicated that HIIT is a potential time-efficient exercise strategy for the treatment of type 2 diabetes. So high-intensity training, if for someone who hasn't worked out, it's going above the 80 percentile of your targeted heart rate. And it is pretty intense, and it needs to be taken slowly to get there. These are some of the data that was collected from the same research. The cellular benefit of exercise. Exercise restores mitochondrial function in type 2 diabetes. The control this is the control group versus the type 2 diabetics. The black bar is for pre-exercise, white bar is post-exercise, and same for the type 2 diabetes. And they measured the phosphocreatine. This also is one thing that reflects the mitochondrial function. So how good it's working in living organisms. And as you could see from the graphs, that post exercise for both the control and the type 2, diabetics group, they both had better function of the mitochondria. These are graphs that shows Glut4 and the citrate synthase. Glut4 is also shown in higher concentration post-workout, hence showing a better glucose clearance. So there was no comparison of before and after it was pre and post of Glut4 concentrations. And as you can see post, you see more Glut4 on the cell surfaces. And citrate synthase is also a mitochondrial marker for its activity. And it is shown increase in the post which indicates better mitochondrial function. This graph basically shows a 24-hour average blood glucose concentration. And you will see that post-exercise, it shows a decrease, which shows that glucose clearance improved post-workout. Exercise recommendation, and these recommendations were from the research paper. Most studies have examined the therapeutic effect of exercise in people with type 2 diabetes, have focused on low to moderate intensity aerobic exercise, which has been established as an effective strategy to improve the metabolic health of people with type 2 diabetes. The recommended amount of exercise for people with type 2 diabetes is 150 minutes of moderate intensity exercise per week. The second research that I'd like to introduce is called Improvement of Insulin Sensitivity in Response to Exercise Training in Type 2 Diabetes Mellitus, is associated with vascular endothelial growth factor A expression. This is the second part of how exercise helps us. The assumptions were that exercise leads to larger endothelial cell surface area, which enhances blood flow to skeletal muscles, which leads to enhanced glucose uptake, which then leads to changes in insulin sensitivity. The second assumption is that in type 2 diabetic patients, insulin-mediated capillary recruitment is impaired, also known as insulin insensitivity. Some of the terms that's discussed in this research is VEGFA, also known as growth factor A. I'll mention it as growth factor A. It's a key molecule in angiogenesis, that is formation of new blood vessels. These cells are specialized and are capable of stimulating cell growth, division of cells, and in processes such as wound healing. MI would be also referred to as insulin sensitivity index. People with type 2 diabetes already know this index basically by checking their blood glucose. These are between 70 to 130 for before meals, the blood sugar that we check before meals and after meals. After 2 hours of eating, the blood sugar glucose level should be 180. So that's exactly what insulin sensitivity index is. The method of this research was 12-week trial with type 2 diabetic mellitus patient who had A1C levels of more than equal to 7.5. These participants needed to be 40 to 60 years of age and with a BMI of 25 to 30. So they weren't normal, there were overweight participants as well. They were on 12-week treatment of diet and 1 glucose lowering medication. The medication was not specified, so we don't know how the glucose lowering mechanism worked. And the participant also had to have no history of exercise. They were given a treatment of acarbose, which is a form of glucose. They were given 50 milligrams per day, 3 times a day for 3 weeks, and 100 milligrams 3 times a day from 4th from the 4th week. So there were 2 groups, and group 1 had to exercise more than and equal to 50% maximum capacity of heart rate for 40 minutes. Group 2 had to exercise more than and equal to 80% of their heart rate in 3 periods of 3 to 4 minutes to only engage large muscle training, which is resistance. So group 1 aerobic exercises, running, cardio, cycling, and group 2 was more resistant training. Muscle biopsies were taken as well. These were 24 to 48 hours after last bout of exercise, and this was to gauge different factors and changes. The results showed that vascular endothelial growth factor A, which is VEGF-A expression, was positively associated with an increase in insulin sensitivity and with a decrease in A1C. Conclusion: growth factor A is a major angiogenic growth factor and increases capillary supply to the skeletal muscle. It enhances the substrate and metabolite exchange, and this would be most likely to the mechanism behind the variation of insulin sensitivity. So since we're talking about vascular endothelial growth, we already know that we have people or patients with type 2 diabetes already have a information that the neuropathy is one of the major thing that happens with the continual increased level of glucose in blood. So it's good to know that exercise helps with the health of the blood vessels. And I would say this is partly, it would help with avoiding amputations and other complications that comes with type 2 diabetes. Conclusion and discussion. Well, through all the research, we have come to know that exercise leads to better glucose clearance from blood and overall insulin sensitivity. Exercise leads to better mitochondrial health, which leads to efficient energy production by body. Exercise leads to better healthy blood vessel, and it aids in the healthy blood vessels aids in the neuropathy and other metabolic functions. Exercise leads to managed glucose level, which means better quality of life. Exercise leads to insulin sensitivity in muscles. These are some websites and research work that I also got information from. And thank you.